So hello everyone, this is Pratham Bhavali and I decode how the world really works. So today in this session we're gonna discuss about CRISPR. So how it is doing today in 2026 and what is the future, how it is, and from we're gonna discuss from each and every aspect in this session about CRISPR. So let's begin. So CRISPR, if we are having a roughly definition, that is, it is clustered regularly interspace short palindromic repeats, which is a programmable genome engineering platform capable of creating site-specific DNA double-strand break and triggering endogenous DNA repair pathway. So, uh, the key distinction is CRISPR edits the information layer of biology, not just downstream molecular effects. So, why CRISPR was a revolution? So, before here we're gonna see before CRISPR and what are the advantages of CRISPR. So, zinc finger nucleases required complex, expensive protein engineering, where large protein inefficient delivery, hard to scale, and random mutagenesis impresses shotgun approaches. And where after coming of CRISPR, what are the advantages? Like RNA programmable, simple to design, change the sequence, change the targets, and it's scalable, can target virtually any gene, multiplexing, it can edit multiple genes simultaneously, and it is efficiency ordered of magnitude cheaper, and it is faster. So CRISPR democratized genome editing globally. Then what is this mechanism? The molecular components we're gonna see first. Guide RNA. So, approximately 20 nucleotide sequences complementary to target the DNA, determines specificity. Minor mismatches can lead to off-target risk also, and is having a case enzyme which is known as SPCase9 is the most common one, and it acts as the molecular scissors. Variant like case 12 and case 13 exist for different target RNA versus DNA. Is having a PAM sequence, which is short DNA motif required for binding limits, where CRISPR can cut it act as a safety lock mechanism. CRISPR's precision is conditional, not absolute. Then here we're gonna see the biology. What is how DNA repair pathway works. So CRISPR itself does not edit DNA; it creates damage. The outcome depends on how the cell repair the break. So NH, which is non-homologous end joinings, which is error prone, leads to random insertion or deletions, useful for gene knockouts. And HDR, homology directed repairs, which are precise correction using a template, rare in adult cells, requires for genes repair. So most human cells prefer NH, making precise repair difficult. So, double strand break, DSBs. Why DSBs are dangerous? Creating a double strand break is a violent event for a genome. So, large deletion occurs to loss of substantial genetic material around the cut sites, chromosomal translocations, approaches fusing of two different chromosomes, oncogenesis potentials, and loss of heterozygosity, which approaches to loss of one parent's allele copy, and P53 activation. DNA damages responses can trigger cell death or select for P53 mutate, which is a cancer prone cell. And CRISPR editing risk is a structural, not just sequence based. So this is how CRISPR case 9 works. If you want, you can pause this video and you can take a screenshot of it so that you can see it after. What are the variation? Base editings. Here we're gonna see the mechanism first. We're gonna see it converts single nucleotide like A, G or C, T without creating a double-strand break. It uses a naked case9 fuse to damage enzymes. What are the advantages? It is significantly lower genotoxicity and it is having certain limitations also. And what are the limitations? The limited scope. Only certain transition mutation can be corrected. And then bystander edits. Nearby C's or A's might also be converted unintentionally. Off target RNA or DNA off target deletion is possible. So what are the variation in this? The prime editing in this we will see. Find and replace for DNA. Prime editing uses a case9 nicase fuse to a reverse transcriptase. It writes new genetic information directly into the target site using a PEG RNA, which is primary editing guide RNA. So it can insert, delete or correct all 12 types of point mutations, and it does not require double-strand break or donor DNA. And the current hurdles are lower efficiency in vivo and larger delivery payload. So it represents next generation clinical CRISPR also. So the delivery strategy, what are the delivery strategy we're gonna see in vivo, ex vivo versus in vivo. So in ex vivo, which is outside the body, cell extracted, edited in lab and re-infused. It is highly controlled and easier to check for errors, and it requires conditioning, which is chemo for space you can say. And standard for blood disease maybe SCD, thalassemia. And in vivo, inside the body, edited delivered directly into patients through IV or injection. One shot cure potential is there. It is harder to target specific tissues, immune risk, and it is mostly used for liver, eye and muscles diseases. Which are most approved CRISPR therapies today are ex vivo only. So the most important is the unmet needs. Over 7,000 known monogenic diseases are there, and there are less than 10% have effective treatment. So CRISPR enable root cause correction, not just symptoms management. It offers potential cures after decades of pharmacological failure. So here is the systemic shift. Why traditional pharma fails here? Small molecules and antibodies cannot fix broken genes. In this protein folding, mutation often cause protein to misfold or degrade before a drug can act. It is having absent enzymes. You cannot inhibit or activate an enzyme that isn't there. And broken pathway. Downstream signaling is permanently disrupted as the source. So CRISPR bypasses pharmacology entirely by fixing the code itself. Here is the economics we're gonna see. Economics disruptions are the revenue models first. It replaces long time drug revenue of chronic treatments mainly with the one-time cure. It destroys recurring cash flow models also. So, and reimbursement requires outcome-based payment models. Insurers pay over time only if the cure persists. And the risk profile as pharma now carries biological liabilities, lifelong side effects rather than just clinical risk. What is the translation? So it is first in human trial risk is there, violation safety assumption where we will see traditional drug trial assumes you can stop the drug if toxicity appears. CRISPR is different. Here performance, genomic alteration cannot be washed out, and no reversible agents are there. Once edited, the change is heritable by daughter cells also. And latency, oncogenic risk is there, which is cancer may take year of decades to manifest itself. So there is no switch off buttons, anything, nothing is there. If you are, once you take the CRISPR, it become your heritable, and it gonna take generation wise. So now we're gonna see a case study which is on sickle cell disease. Here the main problem is sickle point mutation in HBB genes causes hemoglobin polymerization, deforming red blood cells. So the CRISPR strategy here is known as Casgevy. So in this, it target BCL enhances the break of fetal hemoglobin, and it destroys the break, reactivates the fetal hemoglobin here, and then the fetal hemoglobin replaces this faulty adult hemoglobins. So here it is an elegant biology, we can say, and it is a complex clinical execution also. So here we want to see the clinical reality. What is the conditional toxicity it will be? So for ex vivo therapy, the patient's existing bone marrow must be destroyed to make room for editing the cell. So here is the malmobilization, which is highly dose chemotherapy. Busulfan, we will know, wipes out the immune system. It is a very toxic process, and here is the severe risk of it: infertility, almost guaranteed, and secondary cancer, severe infections during neutropenia, and then mortality is there, where treatment related mortality risk is 1 to 5%. The cure itself is life threatening. So what is the impact of off-targeting this? So dependencies are CRISPR might cut DNA sequences that look similar to the target, and gRNA designed how unique is the sequence. Chromatin state, is the DNA open or tightly packed? That aspects we have to see. Cell cycle, what is the cell repairing DNA? That we have to see. The detection challenge here is finding a new unwanted mutation in 6 billion liter of DNA is hard. It is a rare event, we will say. It occurs in less than 0.1% of cells, but can still cause cancer. And the casualty, hard to prove if cancer year later was caused by the edit. So absence of evidence is not equals to evidence of absence. That we have to see. So the long-term risk here is clonal dominance. So even if only one cell acquires a growth promoting mutations, for example, we have seen P53 knockout, the cell can outcompete normal cells. Like selection pressures, the body selects for fetus cells. This time scale, a single clone can expand to dominate the bone marrow over years. The outcome, potential triggers to leukemia, MDS or AML, anything. Historical precedents are like a retroviral gene therapy trial in early 2000s caused leukemia via this mechanisms. So in this we can clearly see here. So if you want to know this, you can take a screenshot out of this. And here we're gonna see the immunology here, the immune complications. What is it? So Cas9 is a bacterial protein from S biogenes. Most humans have pre-existing immunity or antibodies to it because we have all had strep throats. So in this, cytokine release is there, which is systemic inflammation upon delivery. Inefficacy losses, immune system destroys the editors before they works. Exclusions are patients with high antibodies titer may be excluded from trials. The immune system was not designed for genome editors. What are the patient experience? This is a very crucial part. Here, existential anxiety is there. Like, if a living with the knowledge that your fundamental codes has been altered, there is a question: am I still me? Then, identity issues. For diseases like sickle cell that shape identity, community, a cure can lead to a loss of belonging. You will not belong to yourself anymore. So, and uncertainty is their burden of the unknown. Every new ache or pain triggers fear of CRISPR's cancers. So, there are some psychological consequences are also there. And what does the ethics says? Informed consent breakdown. True informed consent requires understanding irreversible risk and accepting unknown future harms also. How do you consent to a risk that science hasn't discovered yet? So this is a very big question. CRISPR's consent is philosophical, not just legal. So here is the governance now. What are the regulatory shifts here we're gonna see? The regulators like FDA, EMA are rewriting the rulebook for gene editing. CRISPR is not treated like a standard biological drug. It is genomic surveillance, mandatory sequencing for patients for years, and 15 years follow-ups they have allowed, which is long-term cancer registries required for all trials participants. Then risk mitigation is strict. It is very strict, stopping rules if adverse genomic events occurs. Then what are the access? The access gaps are like 80% of sickle cell patients live in sub-Saharan Africa, where the current CRISPR therapies requires high-tech transplant units, unable in this kind of regions. This is a harsh reality here. Cure exists where disease burden is the lowest. What is the future? So what is the future direction for CRISPR? The next frontier are non-toxic conditioning, antibodies based clearance, indeed of chemo, in vivo based editing, direct injection to avoid transplant complexity, lipid nanoparticles are used in this, non-viral delivery to reduce immune response. Now in this, AI prediction are also used in to prefer to perfect modeling of off-targeting sites before clinical use. So, and what will be happening in pharma transformation? Genetic custodian, which is pharma companies must become lifelong partners to the patients, monitoring their genomes for decades rather than just selling a pill. Data driven success is no longer measured by scale volume but by longitudinal health outcomes and data management. CRISPR forces pharma into lifelong accountability here. So, in this, editing DNA was the easy part. Living with the edit is the real experiment. CRISPR is a molecule miracle, a clinical gamble, and a societal stress test. So, what do you think of it? So, please mention in the comments whatever it is there, whatever are your thoughts. So, thank you. This was a discussion about CRISPR.